Okay, so welcome to this next video in which we are doing a summary of the adaptive immune response. Okay, so we are first looking at the humoral adaptive immune response, which remember is an immune response that is targeted against extracellular pathogens. Okay, so pathogens that are sitting in the tissue fluid with your cells, but are not actually within your cells themselves. Okay. So, we've just seen how the antigen-presenting cell will search and search and search through the lymph node to find a CD4 positive naive T cell that has a complementary T cell receptor for that antigen fragment. Okay, right. When it finds that, it will cause this CD4 positive naive T cell to become active. Okay, so it will activate it. And when the CD4 positive naive T cell activates, the first thing it does is it differentiates into what's known as a T helper 0 cell or a T helper 0 cell. Okay, often denoted like T subscript H 0. Okay, so after the initial differentiation, what's going to happen is these T helper 0 cell, this T helper 0 cell is going to proliferate and divide and divide and divide and divide and form a huge, great population of identical T helper 0 cells. Okay, so here is our population of identical T helper 0 cells, all of which have this T cell receptor which will bind to this antigen fragment which has been presented. Now, by mechanisms which are not understood, what's going to happen is if the pathogen is extracellular and therefore you need a humoral adaptive immune response, what will happen is most of these T helper 0 cells will now differentiate into what are known as T helper 2 cells rather than T helper 1 cells. Okay, now this process is not understood. We do not know how really the presence of an extracellular pathogen leads to this differentiation into T helper 2 cells. What we do know is that interleukin-4 appears to be extremely important in this, but we don't know where this interleukin-4 actually comes from. Okay, so alternatively, some of these T helper 0 cells may differentiate into memory cells, okay, and what these will do is they will remain within the lymph node, and it will mean that you have a greater population of T cells which have this T cell receptor which is complementary to this specific antigen fragment within that lymph node. So, if you get a secondary infection of this extracellular pathogen, so if you wait many years, let's say, and then along comes this infection again, then what will happen is the same process of antigen presentation will happen. A professional antigen presenting cell will take the antigen on MHC class 2 to the lymph node, and it will again have to search for T cells which have complementary T cell receptors on their surface in order to activate those T cells. This time it will happen quicker because you'll have a much greater population of T cells in that lymph node, and in fact in the body, because the T cells aren't trapped in a lymph node. They can move out of that lymph node and go to other lymph nodes or to the spleen or other lymphoid tissue. Okay, so they aren't bound to a single lymph node. So basically, if you get this second infection with this extracellular pathogen, you'll go through the same process of antigen presentation, only this time it will find a T cell which is has a complementary T cell receptor to this antigen fragment much quicker, and therefore you'll get activation of this T cell much quicker. So the memory T cell, once it's been activated, and interestingly, its activation doesn't require the same level of checking as the activation of the initial CD4 positive T cell. So this was a naive CD4 positive T cell. This had never had this process happen to it before, whereas memory T cells, those only require the antigen presenting cell to bring MHC class 2 with the antigen fragment on. They don't require these other steps here. They don't require the co-stimulatory molecules. Okay, what will then happen is the memory T cell will divide many times, and it will differentiate back into a T helper 0 cell, and those T helper 0 cells will then go through this same process. You'll generate a population of T helper 0 cells with these T cell receptors which are complementary to the antigen fragment far quicker in your secondary immune response. Okay, and that's why the secondary immune response is generally much stronger and much faster than the primary immune response, and generally why the infection when you get infected for the second time, generally it can't establish a big enough hold on you to actually make you ill. Too quickly the immune system will become armed against it and will eradicate it, basically. And that's the basis of vaccination, that we will expose you to the pathogen in a non-dangerous form. The immune system will mount an immune response to it. You'll get all sorts of memory cells, and then if you ever actually do get infected with the pathogen, then the immune system will mount a secondary immune response rather than a primary immune response, which will be much quicker and much stronger than the primary immune response, and will eradicate the pathogen before it can get any sort of a hold on the body, basically, and therefore it will prevent you from actually getting ill. Okay, because it can't get a big enough hold on you to actually lead to becoming ill. Okay, right. So what are the T helper 2 cells actually going to do then now? Well, basically they are involved in helping B cells. So basically we've discussed that when the pathogen, when this extracellular pathogen is present in your body, all sorts of antigens will be released from that pathogen. Maybe the pathogen will actually be secreting or releasing them into the interstitial fluid itself, or maybe the pathogen will be being phagocytosed by neutrophils, and the neutrophils will be releasing these antigens after they have phagocytosed the pathogen. Okay? Either way, what is happening is that there will be many of these antigens in the lymph that is coming to the lymph nodes. Now, the B cells that are in the B cell follicles of the lymph node will be being exposed to these antigens, which have to percolate, remember, through the cortex and the paracortex to get to the medullary sinus. Okay? Now, just like with T cells, all B cells will have a slightly different B cell receptor on their surface, which will have a slightly different antigen binding site here. Okay? So, what will happen is if you have a B cell within a B cell follicle which has a B cell receptor which is just right for binding to this antigen, then it will bind to this antigen, and then what will happen is it will internalize the B cell receptor, break down the antigen, and express fragments of it on its cell surface with MHC class 2. Okay? Then it will also, because it's being exposed to all these antigens, some of them will be PAMPs, basically. Some of them will activate pattern recognition receptors within this B cell. So the B cell, which is another type of professional antigen presenting cell, will then present on its surface CD4 and B7.1, B7.2 after activation of its pattern recognition receptors. Okay, so that's very important. Only professional antigen presenting cells can be induced to put these co-stimulatory molecules on their surface. What will then happen is the B cell will find this T helper 2 cell, which has the complementary T cell receptor for this antigen fragment here. So let me just label this thing up in red here as the T cell receptor, the TCR. Okay, so when this happens, when the B cell finds this T helper 2 cell, then the TCR will bind to the antigen fragment mounted on the MHC class 2. The CD4 on the surface of the T helper 2 cell will bind to the MHC class 2. The B7.1 or B7.2 will bind to CD28, and CD40 will bind to CD40L on the surface of the T helper 2 cell. And this is what's known as T cell help. Okay? And it will activate that B cell to divide and divide and divide and divide. So it'll produce a whole colony of B cells that are based on this B cell, and that produces a new little population of cells within the B cell follicle that's known as a germinal center within that B cell follicle. Now, these B cells will not all be genetically identical to this B cell. Instead, what happens is a process known as somatic hypermutation, where all of these offspring here will undergo slight mutations in their B cell receptor antigen binding domain, so that you have variation in this population that you've just generated. Okay? Now, some of them will undergo mutations that mean that their B cell receptors are less good at binding to this antigen, whereas some of them will undergo mutations that mean that they are better at binding the antigen. Okay? Now, basically, in order to survive, the B cell must have a B cell receptor that binds the antigen well, because there is a limited amount of antigen. So in these B cell follicles, there is a type of cell known as a follicular dendritic cell, which is binding to the antigen that is percolating through the cortex and the paracortex of the lymph node, and is holding it on its cell surface membrane so that the B cells are continuously exposed to this antigen. Okay, but even still, there's still only a limited amount of antigen available. So the B cells which have the best B cell receptors, which are best at binding to these antigens, they will get the antigen, and those which don't have a good receptor, those will fail basically. So the ones which fail to bind antigen, they'll end up dying, and the ones that do, they'll commit suicide, they'll go through apoptosis. Okay, whereas the ones which do bind antigen, what they do is they go through this process again. So they internalize the B cell receptor with the antigen bound to them, they chop up that antigen and present fragments on the surface bound to major histocompatibility complex class 2. Okay, they will then also have the co-stimulatory molecules, and they can go back, find a T helper 2 cell, and interact with it and get T cell help again. And then what will happen is this one now, here, that was good at binding the antigen, that competed, basically, that was selected for because it had a good B cell receptor, it will now divide over and over again, basically, to make a whole population of B cells that are based on it. And in this population, again, they will undergo the process of somatic hypermutation. Okay, so you'll have a whole bunch of variants which are all based on this B cell now. Okay, and again, you'll go through this selection process to select out the B cells which have a B cell receptor that is best at binding to the antigen. You continue this process over and over again, and what you gradually do is you select for B cells which have B cell receptors which are best at binding to the antigen, and the B cells which don't have don't have B cell receptors which are good at binding to the antigen, or maybe they are good but they're just not good enough to compete with these even better B cell receptors, those will undergo apoptosis because they won't get antigen. Okay, and then when they don't get antigen, they can't present it on MHC class 2. They can't present fragments of it on MHC class 2, and they can't get T cell help, and without T cell help, the B cell will not survive. So with T cell help, it'll divide and divide and divide, but without it, it'll just undergo apoptosis. So the T cell help not only promotes the survival of the B cell, but also causes the proliferation. Okay, right. So this process leads to you gradually creating B cells with a better and better B cell receptor as far as binding to this antigen is concerned. Okay, now it's not clear what leads to an end in this process, because this process could go on forever basically. However, something happens. The B cells seem to eventually get fed up, and one of these fantastic B cells that you have now selected for will eventually start dividing and dividing and dividing, and this time it'll produce a bunch of cells which will become plasma cells basically. Alternatively, some B cells will differentiate into memory B cells rather than plasma cells. Okay, so let's now discuss plasma cells and memory B cells, but we'll do that in the next video.